next products are antibiotics. The antibiotics produced by a different bacteria and fungal members are also here antibiotics. Antibiotics. What is antibiotic? It is derived from the Greek word. The Greek word anti means against, bios means life. So these two are Greek words. So what is the meaning of antibiotic? Against the life. So against the life means it is retarding the growth of some microbes which are disease causing. So the disease causing organisms or microbes are killed by these antibiotics. So why they are not killing human beings or other animals? So antibiotic, it is against the life. It is killing only bacteria, harmful bacteria and pathogenic bacteria or pathogenic fungal members. Okay, here these antibiotics are generally they kill only harmful pathogenic organisms. They are not harmful to human beings and they are not harmful to plants or animals. So here antibiotics are only harmful to they are killing the harmful pathogenic bacteria or microbes. So these are some microbes are targeted. So these targeted harmful pathogenic organisms only killed by these antibiotics. They are not harmful to human beings or non-targeted animals, non-targeted organisms, not harmful to host. So in diseases we are taking so many antibiotics. The antibiotics are killing only microorganism which is harmful to us, but not killing us. Why? Because it is targeted. The antibiotic is made on some processes. The antibiotic can kill only pathogenic bacteria. Here the how it is killing only pathogenic bacteria. So by inhibiting the different type of process, like here how is the function? What is the function of antibiotic? Antibiotics function, how they are killing the pathogenic bacteria or microbes. The function of antibiotics. Antibiotics they inhibit the protein synthesis, 30S, 50S ribosomal synthesis and cell wall synthesis inhibition. Cell wall synthesis inhibited or inhibition. (30S, 50S ribosomes ribosomal subunits synthesis is inhibited. Next, protein synthesis. 30S, 50S means the ribosomal subunits. Ribosomes are the protein factories. They are synthesizing the proteins. If the 30S, 50S subunits are not synthesized, no protein synthesis. So protein synthesis is also inhibited. Sometimes plasma lemma synthesis. Plasma lemma synthesis is inhibited by the antibiotic. The pathogen or bacterial, fungal members are generally they are having some cell wall. The cell wall synthesis is inhibited, and sometimes inside the microbe the protein synthesis is also inhibited. So, by inhibiting the cell wall protein synthesis, they are killing or retarding the microbial growth. Alexander Fleming discovered the antibiotic penicillin. The penicillin antibiotic was discovered here unexpectedly. So, while he was studying Staphylococcus bacterium, he was studying the Staphylococcus bacteria. So, in the petri dish, the Staphylococcus bacteria are there. So, he was studying about the Staphylococcus bacteria. In unwashed petri dish, some bacteria are remaining. So, in unwashed petri dish, the bacteria are also growing, but in some places no bacteria were observed. Why? Because here total bacteria are there. Other bacteria, but in some place here in this area no bacteria are observed. So, Alexander Fleming observed some fungal member growth in unwashed petri dish. So, this fungal member later identified as Penicillium. So, Penicillium is a fungal member, it is growing in the unwashed petri dish. So, in unwashed petri dish all the bacteria are growing except in one area. In this area no bacteria are growing. Why? Because the Penicillium is there. So, the Penicillium is inhibiting the growth of other bacteria. So, Penicillium fungus member it is inhibiting the growth of bacteria. So, here Penicillium is working like an antibiotic. It is killing or it is inhibiting, it is retarding the growth of bacteria. That is why here the Penicillium, from the Penicillium fungus one chemical is extracted, later it is named as penicillin antibiotic. First penicillin was extracted by Alexander Fleming from Penicillium notatum. First it was identified by Alexander Fleming in Penicillium notatum. Next here extensively or industrially in large quantities it is extracted from Penicillium chrysogenum. Discovered by Alexander Fleming while he was studying Staphylococcus bacteria. He observed one fungus growth, it is inhibiting the growth of other bacterial members. Here no bacteria are present in this. Here no bacteria are growing in this area. Why? Because the Penicillium fungus member is there, that is inhibiting the growth of bacteria. So, first he identified in Penicillium notatum the penicillin antibiotic. It is extensively extracted from Penicillium chrysogenum. Nowadays we are taking penicillin antibiotic. This penicillin antibiotic is industrially produced from the Penicillium chrysogenum. Next antibiotic is streptomycin. Streptomycin, it is an antibiotic extracted from Streptomyces griseus. Bacterium, so penicillin, it is penicillin antibiotic extracted from fungus Penicillium notatum or Penicillium chrysogenum. Here streptomycin is also one antibiotic. It is extracted from Streptomyces griseus. It is a bacterium. So, number of antibiotics are extracted from here Streptomyces species. Here nowadays 8,000 of antibiotics were identified. 8,000 antibiotics were identified or discovered. In this here more than 41 antibiotics are here from Streptomyces species. Here Streptomyces species and 60 antibiotics, almost 60 antibiotics are extracted from Bacillus bacterium, Bacillus species. So, Streptomyces, it is producing streptomycin, an antibiotic, and retarding the growth of pathogenic harmful bacteria or fungal members. So, here two types of antibiotics are there. One is broad spectrum antibiotics, and the second one is specific or narrow spectrum antibiotics. Broad spectrum means here the antibiotic is killing different type of organisms. It is pathogenic. The pathogenic microbes are killed by these antibiotics. Here all these are broad spectrum antibiotics. These are killing the number of pathogenic microbes. Different groups of pathogenic organisms are killed by these broad spectrum antibiotics. Here chloramphenicol. It is extracted from bacteria Streptomyces venezuelae. It is also known as chloromycetin. Streptomyces is a actinomycetes bacterium, and chlorotetracycline, also known as aureomycin, extracted from Streptomyces aureofaciens. It is also actinomycetes bacterium, and oxytetracycline, also known as terramycin, also extracted from Streptomyces rhinosus. Next, fumagillin. It is extracted from Aspergillus fumigatus. The fumagillin is extracted from Aspergillus fumigatus. It is a fungus. All these are bacteria. So, these broad spectrum antibiotics kill different type of organisms which are causing the diseases. They may be bacteria or some parasites or other organisms also. Narrow spectrum antibiotics, they kill only some specific pathogenic organisms. So, only one type of or a few types of bacteria or pathogenic organisms are killed by these narrow spectrum antibiotics. So, broad spectrum they can kill different type of pathogenic organisms, but narrow spectrum antibiotics can kill only one specific or a few specific pathogenic organisms. So, these are some narrow spectrum antibiotics. Penicillin, already as we discussed, the penicillin is extracted from a Penicillium notatum or Penicillium chrysogenum. It is a fungus. Next, streptomycin. Streptomycin is it is extracted from Streptomyces griseus. Streptomyces griseus is a bacterium. It is actinomycetes bacterium. Next, erythromycin. Erythromycin is extracted from Streptomyces erythraeus. It is also one actinomycetes bacteria. Polymyxin B. Polymyxin B, it is here extracted from Bacillus polymyxa. It is here spore producing or endospore. In some bacteria, endospores are formed. So, Bacillus polymixa is a endospore producing bacteria. Endospore producing bacteria. Streptomyces, the species Actinomyces bacteria. Penicillium is fungus. So, penicillin, first antibiotic, it is first discovered by Alexander Fleming. Streptomycin, it was observed, identified and extracted by Waxman scientist. Streptomycin was extracted by Waxman scientist. For these, for the discovery of penicillin and for the discovery of streptomycin also, these scientists got Nobel prizes. Penicillin, for the discovery of penicillin, Alexander Fleming and Florey Chain scientist. These three scientists got Nobel prize. For this discovery, Alexander Fleming, Florey and Chain scientist. So, three got Nobel prize in 1945. Next, for the discovery of streptomycin, Waxman scientist. Waxman scientist got Nobel prize. So, these are some antibiotics. Antibiotics are against the life, and these target is some pathogenic fungal or fungal members or bacteria or other pathogenic organisms, and these are extracted from bacteria or sometimes fungal members also. And here these are not killing the non-target organisms. So, the antibiotics are of different types, 8,000 of antibiotics, and these antibiotics are most of the antibiotics are extracted from Streptomyces species and Bacillus bacterium. So, these are of two types, broad spectrum and narrow spectrum. Broad spectrum can kill the different type of organisms which cause the disease. The narrow spectrum, they are specific. Narrow spectrum also called as specific antibiotics. These specific antibiotics can kill only one type or a few types of pathogens. So, it is about antibiotics.